Welcome everyone to today's Explainer. Today, we're taking this absolutely incredible spatial journey straight into the microscopic building blocks of literally every single living thing on Earth. You know, it's so easy to look around at humans, towering trees, or even simple single-celled organisms and see them as completely different entities. But when you strip all of that away, when you magnify the very fabric of our existence, we are all constructed from the exact same fundamental unit. We are talking about the cell. I am super thrilled to be your guide as we navigate through this microscopic universe, and I promise you, by the end of this explainer, you are never going to look at a living thing the exact same way again.
Okay, so here's the roadmap for our explainer today. We'll start off with a microscopic discovery. Then we'll ask the question, one cell or many? After that, we'll meet the cellular gatekeepers. Then we'll pierce right through to the control center. And finally, we'll take a tour inside the cell factory.
Section 1. A microscopic discovery. Okay, let's dive into this. Because to really understand the cell, we actually have to transport ourselves all the way back to the 17th century. Imagine a world where the idea of microscopic life was, well, pure science fiction. Literally no one knew what made up living tissue. Then, this totally chance observation completely changed our understanding of life forever. In 1665, an English scientist named Robert Hooke took a really thin slice of cork, which is literally just tree bark, and popped it under this primitive, self-designed microscope. He didn't see a solid block of wood. Instead, he saw this totally wild, honeycomb-like structure made up of all these little separate compartments. And because these tiny boxes kind of reminded him of the small, bare rooms where monks lived, he coined the Latin term cells, which translates to a little room. It might seem like this small, insignificant incident, but this was the very first time in human history that someone observed that living things consist of separate, distinct units.
And Hooke's seemingly tiny observation, it sparked a super rapid scientific timeline. Just a few years later, in 1674, a guy named Leeuwen Hooke used an improved microscope to discover actual free-living cells swimming around in pond water. Then, zooming ahead a bit to 1831, Robert Brown discovered the nucleus hiding inside the cell. And this whole thing culminated around 1838 and 1839 when biologists Scheiden and Schwann presented the foundational cell theory. Basically, they proposed that all plants and animals are composed of these exact same basic units of life. This was later expanded to include the absolutely crucial point that all cells actually arise from pre-existing cells. We went from a piece of cork to a unified theory of biology. How cool is that?
Section 2. One cell or many? The way cells are used to build organisms actually varies wildly. We're talking everything from simple, independent unicellular organisms just living their best solo lives to incredibly complex multicellular bodies like yours and mine, where literally trillions of cells collaborate every single second. And this brilliantly illustrates exactly how that works in complex bodies. Check out that incredibly elongated, branching human nerve cell. Its whole job is to rapidly transmit signals across really long distances in your body, hence that long, wire-like shape. Now compare that to the other shapes. You've got these spherical fat cells, or those small, disc-like blood cells designed specifically to squeeze through tiny capillaries. The absolute crucial point here is that a cell's shape and size are strictly and directly dictated by its specific function. There's this brilliant division of labor, where different shapes do very different jobs, but they're all working together just to keep you functioning. Section 3. The cellular gatekeepers. So, we know cells come in all sorts of crazy shapes, but let's shrink ourselves down even further now and approach the outer boundary of a single microscopic cell. You can kind of think of a cell like a super exclusive factory. You can't just have random stuff flowing in and out all day. You need gatekeepers to manage the inventory, and this is the selectively permeable plasma membrane. Oh, and fun fact, in plants, fungi, and bacteria, there is an additional rigid outer layer called the cell wall. Now, one of the absolutely most important jobs of this plasma membrane is managing water. The membrane lets some materials pass through while totally blocking others. Water obeys the special rule of diffusion called osmosis, which is really just the net diffusion of water across a selectively permeable membrane toward a higher solute concentration. In plain English, water naturally moves to where things are more concentrated, basically trying to balance the whole system out. Let's anchor this abstract idea in reality with a fun little kitchen experiment. Imagine you dissolve the hard shell off an egg using some dilute hydrochloric acid, so you're left with just that thin, semi-permeable membrane holding it all together. Drop that egg into pure water, what we call a hypotonic environment, and water is going to rush in to balance out the higher concentration of stuff inside the egg, making the whole thing swell up.
But if you take a similar deshelled egg and pop it into heavily concentrated salt water, a hypertonic environment, water rushes out of the egg into that salty solution, and the egg totally shrinks. And hey, if the environment perfectly matches the inside of the cell, which is an isotonic environment, the water moves evenly in and out, and the cell stays the exact same size. So this is exactly how our cellular gatekeepers manage the life-sustaining flow of water.
Section 4. The Control Center. All right, let's leave the gatekeepers behind. Moving deep into the very core of the cell, we find the vault where the blueprints of life are actually kept. We're talking about the nucleus. But as it turns out, not all control centers are built the same. We actually divide life into two major categories based on their nucleus. Eukaryotic cells, like the ones in plants and animals, are super complex. They have a well-defined nuclear membrane, which is this double-layered vault that protects our chromosomes, DNA, and genes. But then, on the flip side, we have prokaryotic cells, like bacteria. The word prokaryote literally translates to primitive nucleus. These guys don't have a defined nuclear membrane at all. Instead, their genetic material just kind of hangs out loosely in an undefined region called a nucleoid.
Now, what's really interesting about this slide is seeing exactly what this primitive structure looks like. There is absolutely no dark, defined spherical nucleus here. It's just a tangle of genetic material floating around. Plus, this highly streamlined cell lacks most of the membrane-bound organelles that complex cells use to separate all their different chemical activities. It's an incredibly stripped-down version of life, but man, is it highly effective.
Section 5. Inside the cell factory. Now, if we step out of that secure nucleus in a complex eukaryotic cell, we enter the fluid cytoplasm. And this space is completely packed with highly specialized, membrane-bound organelles. If the nucleus is the management team, these organelles are the living, breathing machines right out there on the factory floor. One genuinely fascinating piece of machinery is the Endoplasmic Reticulum, or the ER. It looks like this huge, complex network of membrane-bound tubes and sheets spread all throughout the cytoplasm. And it serves as both the primary transport channel and a major manufacturing hub for the cell. And it comes in two distinct flavors. The rough ER looks bumpy under a microscope because it's literally studded with ribosomes. Those are the tiny machines that manufacture vital proteins. The smooth ER, on the other hand, lacks those bumps. Instead, it completely focuses on manufacturing fat molecules and lipids, some of which are actually used to build the cell membrane itself. Another absolutely crucial piece of machinery is the Golgi Apparatus, which gets its name from its discoverer, Camillo Golgi. And let me tell you, the story behind this is just fascinating. Golgi was an Italian medical officer back in the late 1800s. He started his profound microscopic investigations into the nervous system inside a little hospital kitchen that he had basically just converted into a makeshift lab. Working right there in that kitchen, he invented a completely revolutionary technique using a weak solution of silver nitrate to stain individual cell structures, a method he famously called the black reaction. This makeshift kitchen science was so groundbreaking that it finally allowed us to see these super delicate cellular details for the very first time. In fact, it was so huge that it eventually won him a Nobel Prize in 1906.
It's honestly just mind-blowing what we can find when we simply look closely enough. I mean, we've gone from Robert Hooke peering at tree bark and seeing little rooms to Camilla Golgi's staining tissues in a hospital kitchen, all the way to our incredibly detailed modern understanding of the complex factories humming away inside of us right this very second. From a single free-living organism to the trillions of specially shaped cells collaborating in our own bodies, the fundamental unit of life is an absolute masterclass in microscopic engineering. As our microscopic technology gets better and better, I want to leave you with this provocative thought. From a single cell to complex human bodies, what other microscopic secrets are still waiting to be discovered deep inside of you? Keep wondering, keep learning, and I'll catch you on the next explainer.